Mental health problems are rapidly increasing in young people. I mean, there's some evidence from recent surveys of that going up. Approximately 1 in 4 17 to 19-year-olds, from 1 in 6 over a few years ago.
Early life stress exposure or adverse childhood exposure is one of the strongest risk factors for all sorts of psychiatric disease, including depression. What is not known is exactly what's underlying this phenomenon. So I've developed a stress model using zebrafish. Zebrafish being small, we are able to sort of figure out every single gene that is changing. We look at the entire brain and figure out all the changes that are occurring.
Understanding RNA abundance and diversity is a really important factor in discovering new treatments and diagnostics. We study transcriptomics using long-read sequencing technology, which is a novel and next-generation sequencing approach. We are really excited to have discovered thousands of RNA molecules across the developing human brain and in young people. And now we're thinking about the implications in neurodevelopmental disorders such as autism and schizophrenia.
It's really about trying to improve interventions for people at young ages to prevent and improve response to treatments, using neuroscience as well as experimental studies to understand the mechanisms of how that works and then use that to derive better treatments. Is we've actually developed treatments that are effective at reducing rumination and worry and overthinking.
In order to find therapeutics for depression and other mental health related conditions, we need research in two areas. On one hand, we need a very strong research using human patients to realize essentially exactly the conditions and then problems that these patients have. On the other hand, in order to find therapeutics, we need to have a very deep understanding of molecules and the individual neurobiological processes, and that requires a fundamental basic research.
The University of Exeter is leading the way in this research because we have developed a critical mass and a philosophy that involves linking across different methodological approaches. So the work that we do is really informed both by doing clinical trials where we test interventions, but also by experimental work in the lab where we can understand a bit more about the mechanisms of how things work. So for example, one of the studies we've been running has been a trial of a specifically adapted form of cognitive behaviour therapy in adolescents, and we've been using fMRI brain imaging before and after the intervention, and we've seen changes in patterns of brain activations.
We're really excited to extend this work into spatial and single-cell transcriptomics, and also to further analyse the effect of all these RNA molecules on proteomics.
We really want to make sure we're making a huge difference in improving mental health for young people, improving interventions, improving prevention. So we've done a lot already, but there's a lot more to be done.
A dream will be to be able to find the molecule or the regulatory mechanism that we can tweak around, that we can find drugs for, drug target. And it would be that if we can understand that there is this one master regulatory mechanism that links these processes, early life stress and then adult or adolescent dysfunction, then we can think about tweaking that or changing that using a drug, and that can be a therapy.